Okay, so this is where we left it last time. I just gone over sort of how to fill in a Punnett square. When you're trying to predict the outcome, for something like a single gene cross like this, especially if you've done Punnett squares quite a bit before in other courses, you may not need to do them for this kind of work, but you may find them very helpful when we're talking about two gene crosses and things like that, and when we're thinking about genes that are linked on chromosomes. Okay, this is named after Punnett, the person who kind of invented this way of um, laying out what the outcome of a cross is likely to be. And this is how you would answer, if I said, here's these genotypes, we cross them to each other, what are the outcomes? You would use a Punnett square to predict those outcomes. So for homework, I will take this off of your slides. These are basically every possible combination of big A, little a, big A, and so on. And it shows you what the outcomes are in this final section of the table. And so the homework for you is to draw each one of them on a Punnett square as practice and just convince yourself that those are the correct ratios. And if you don't get the ratios that are shown in the table, then there may be an adjustment needed in the way you're approaching the Punnett square, and you can just talk to me or I was going to say one of the T's, talk to Emily, your TA, and we can help you sort that out. Okay, monohybrid and test crosses. This is just terminology. And individuals from pure breeding lines that are homozygous, individuals that carry two different alleles are called heterozygotes, right? And so if you have, if this was your P generation, your homozygous ones, and your F1 are the heterozygous one, if you cross the individuals that are heterozygous to each other or allow them to self-pollinate, as we can do with plants, this is referred to as a monohybrid cross. So the word hybrid here basically means the same as heterozygous, and mono means one gene. So this is crossing individuals that are heterozygous for a single gene, and this is the kind of cross that will give you that 3-to-1 ratio. Okay. Now, a cross between an individual that has the dominant phenotype, but their full genotype is not known. We don't know if they're homozygous or heterozygous. Cross to a homozygous recessive individual is called a test cross, or more correctly, a monohybrid test cross. And that's because you can use the outcome of that cross to find out if this individual had been homozygous dominant or heterozygous, right? Based on what the outcome is. Now, when we use the term monohybrid test cross, except if I phrase it, question differently, this is the cross I will be talking about. So when I say test cross, I'm usually talking about heterozygous individuals crossed to homozygous recessive individuals. And so this is the reason why it's called a test cross, is that Mendel used this cross to demonstrate that the F1 were actually heterozygous, that they had the dominant particle, as he called it, or factor, and they had still the recessive factor. And if they did have one of each, you would expect a 1-to-1 ratio in the progeny, and that is what he observed. But we could use it in other, you found this plant, it's got a dominant phenotype, and you would like to know if it's homozygous or heterozygous, because you want to maybe, if it's homozygous, you could self-pollinate it, and it could be pure breeding, and you could establish a population of pure breeding plants for whatever reason. And so if the individual is homozygous for the dominant allele, then we expect all the progeny to be have the dominant trait. So the phenotype would be all like dominant A, whatever that is. And so basically, if we do a cross like this and we got 20 progeny and they were all the dominant, we would assume it was probably homozygous for the dominant. But you can never be 100% sure unless you look at really a lot, like maybe 100 progeny, okay? Because there's a small but non-zero chance of an individual being heterozygous and the first 20 progeny you look at all have the dominant phenotype, for example. But when we're doing the other cross, this one, all we need to see is one individual that has the recessive phenotype. And if it's the first individual we see, we know that this parent was heterozygous, because the only way you can get that recessive individual is if it got a recessive allele from one parent and the recessive allele from the other, right? So this one is much more certain. We could look at 5 or 6 progeny, and if we see a couple that have the recessive trait, then we can be quite certain of what the genotype of this parent was. Whereas here there's more uncertainty. And the reason is the presence or absence of this recessive trait. Not seeing the recessive trait doesn't mean it's not there, whereas seeing it means it is there, right? And so here it's more a case of the large number of progeny. Every time you see another individual progeny that is dominant, has the dominant phenotype, it makes it less and less likely that this individual was heterozygous, but the probability is never 0, okay? But within, I mean, honestly, if you looked at 50 progeny and they were all showed the dominant phenotype, you'd pretty much be justified in saying, you know, it's homozygous dominant. Okay, and that is, this is just an illustration of that. So Mendel made the prediction that if these ones had both of the factors and he did this cross, he would expect to see half of the progeny with the green phenotype and half with the yellow phenotype. And that is exactly what he saw. And so he got, that was as I said, good scientists always figure out, okay, I've got this idea, how am I going to test it? And if it doesn't come out this way, I have to discard that idea and come up with a different hypothesis, right? But he was also able to use self-crossing of the F2 as another test, right? So he crossed the F1, and remember he got a 3-to-1 ratio, so 75% of the progeny were yellow, 25% of the progeny were green. And within these, he had some that had the 2 factors and some that had just homozygous for just 1, right? And so if we look at these yellow peas, how do we know which are the homozygous ones and which are the heterozygous ones? If he wants to test his hypothesis and say, okay, well, 2 of these must be heterozygous and 1 of them must be homozygous, how's he going to demonstrate that? How can he actually tell which is which? He can't. But what he can do is he can allow each of the plants that comes from, because this is a pea seed, you can make a whole plant out of it, and then you can allow each one to self-fertilize. And the ones that are homozygous are going to be pure breeding. All the progeny will be yellow, but the ones that are heterozygous, you're going to end up with a 3-to-1 ratio. Okay, so that would be the prediction. And that's basically what he saw. So when he allowed those F2s to self-fertilize, he found that 1/3 of the plants gave rise to all yellow. 2/3 of the plants, the yellow plants, gave rise to a 3-to-1 ratio. So he could then go back and claim, okay, those were heterozygous ones and these were homozygous ones. And then, of course, if you allow the green F2 to self-fertilize, of course, they're homozygous, right? So you're going to get all green progeny. And so this was how he was able to add to the confirmation. Every time he made a prediction and got results that confirmed it, then that made his hypothesis more trustworthy, more believable, and so on. And we will go through, this part can be really hard to understand, and it will be important when we look at probabilities, looking at pedigrees, when we look at human traits. And so I will go through a little exercise in the class to help you with this part. And that's it. The next lecture is going to be about autosomes and sex chromosomes. And we'll talk about how the segregation of those alleles differs a little bit. And hopefully you'll find it interesting.